Good afternoon. I'm Jeff Blumberg, and I'm the director of the Antioxidants Research Lab, and it's a real pleasure for me to introduce Liz Johnson, the seminar speaker today. Liz did her undergraduate and graduate training in nutrition sciences at the University of Wisconsin, and another alumni, and then promptly came to the HNRCA for her postdoctoral work and stayed working primarily with Rob Russell, past director of the Nutrition Center here, for almost exactly 27 years, during which time she's done some seminal work on the role of carotenoids in our diet and health. She's also advised or trained about 30 students in 27 years. Liz is very active outside in the nutrition science community. She's served in various capacities on CARIG, the Carotenoid Research Interactive Group. Thank you, I just forgot what that stood for a minute. And as a scientific advisor to ILSI, the International Life Sciences Institute, North America. And she had just last year received the Mark Bieber Award for serving as a wonderful example of bridging between academia and industry by the American College of Nutrition. And we're very fortunate that Liz has decided to come back to Boston after just returning from Korea, and just before that, just returning from Brazil, so she can deliver this seminar. Liz?
Thank you very much, Jeff. That was a kind introduction. And if any of you have been doing your math, you'll realize that 2014 minus 27 is not 1988, which is when I got here. Actually, this month, 27 years ago, was the first time I entered this building, and it was just for a visit. I didn't know anyone here, so I knocked on Rob's door, the associate director at that time. I said, I'm Liz Johnson. I know about vitamin A. I know about HPLC. And he said, do you want a job? And honest to God, it was that simple. So I've been here ever since, and afterwards I asked him if he wanted to see a resume, and he said sure. So I've been there since, and it's been great, and you'll hear a little bit about what I've been doing these past 27 years. This doesn't seem to work. All right, so this isn't working. Point to the computer?
Just try the
It doesn't
manual.
work.
How about this one?
So anyway, this is what I do. I work on these things. I work on carotenoids that we find in our fruits and vegetables, and the one that I started out working with was beta-carotene, which you find in your carrots, your pumpkins, and now I'm working on, So anyway, okay. So anyway, I came here in 1988, and the first thing I did was I got involved with the metabolic research unit. So my study then was involved with beta-carotene, because at that time, that was the carotenoid of interest, because among all these hundreds of carotenoids, beta-carotene was the one that was major to our diet, and it had the most pro-vitamin A activity. So everyone wanted to know about beta-carotene, and so that's what I did. So here I am, coming from Wisconsin, home of Bucky Badger, 1988, working with Rob Russell, who at that time was the associate director, and involved with the metabolic research unit. And what we did, we just wanted to get some descriptive work on beta-carotene and its absorption and how it travels throughout the body and the lipoproteins. So we did that, and what we found was when you gave this big whopping dose of beta-carotene to these people and looked at their plasma appearance in plasma to get an idea of absorption, there was a number of people who had no response, and we called those non-responders or low responders. So this was really interesting, is why are some people different in terms of how they absorb the beta-carotene? And this theme will come up later when I talk about another carotenoid, but that's something we described early on. So then I spent more time working on some descriptive data on what's going on in humans and working with the MRU. And we looked at sex differences in absorption of vitamin A. And the interest for vitamin A was because of beta-carotene. And we found that women are different than men in terms of absorption, and where their postprandial clearance of vitamin A was faster, so tissue uptake was a little bit better in women. We also worked at different forms of vitamin A in people with GI disease and pancreatic disease, and to no surprise found that more water-soluble forms of vitamin A were better absorbed in people who had GI problems. And so if you look at any of my protocols that I hand into MRU, that's always an exclusion for anything I do as far as the carotenoids go. So that's the stuff I was doing in the GI nutrition lab. And then I stepped out of that lab, and the first collaboration I had was with Bess Dawson Hughes and Jerry Dalal, where we looked at the effect of vitamin A supplementation on measures of liver function. And the reason for that is that at this point in time, we had these vitamin A supplements that have been around for a while, and there was this potential to cause toxicity because vitamin A is fat soluble. You can store it, and with chronic high intake, there's potential for toxicity. And so that's what we did in this study, and we found, in fact, that there really was no measures of liver damage in those who use vitamin A supplements. So that was my first collaboration here. And then I started doing work with Norman Krinsky. And I'm not sure if any of you probably you young ones may not know who he is, but for me at this point in time, he was like the guru of carotenoids. This man, he had been studying carotenoids longer than I had been alive. In fact, his PhD dissertation on the biological effects of carotenoids was handed in, and he got his PhD the very year I was born. So he had been studying this forever, and he was someone who first recognized that carotenoids did something more as far as biological effects than being pro-vitamin A molecules. So he recognized that they have potential for human health. So this was really terrific, and he became a mentor, a colleague, a friend, and that was really a big highlight for me to be involved with him. And I did some work with Shang and him as well. And in this study here, this was really exquisite work that Shang did with his surgical skills, where he looked at the ferret and did some cannulations of the lymph, where we could look at differences between two isomers of beta-carotene, the trans and the 9-cis, and look at their metabolites, because at this time, people were realizing that these forms of retinoic acid, 9-cis trans, came from beta-carotene, but were they coming from these isomers of beta-carotene? So Shang was able to do that. I was lucky to be involved with that. And something about this photo, I want to tell you, this photo was actually taken a few months before Norman died. And this photo was at his birthday party because he knew he was going to die, but he said, I want to have a party. So this was at that, and he really was an inspiration. He really was really a big, important part of my research early on. So, okay, getting back to 9-cis retinoic acid, 9-cis beta-carotene. This was work that we did in animals, but almost all my work that I've done has been with humans. So we wanted to look at what was going on as far as distinguishing if there was a difference between absorption of trans beta-carotene and 9-cis beta-carotene. So these are two different carotenoid isomers of beta-carotene that have different biological effects because of their metabolites. So the work we did with Shang found that yes, 9-cis beta-carotene will turn into 9-cis retinoic acid, which have different biological effects than the trans form. But that was in ferrets. What's going on in people? And so in this study, we looked at these isomers of beta-carotene, and we wanted to see if there were differences in intestinal absorption and tissue uptake. But I wanted to look at it at people. And so this was in 97 that we published this. But in 1990, 1992, and in 1996, I had 3 boys. 3 boys that I was breastfeeding. And I remember sitting at the HPLC and thinking about, okay, I gotta, bring home some milk tonight, and realizing that my milk was yellow. And my milk was yellow because I like carotenoids. And I thought, wait a minute, these carotenoids from my diet are getting absorbed and appearing in my milk. I said, I have a marker of tissue uptake if I just analyze breast milk. And so that's what we did. And I worked with Leche League, and we went out and got breast milk, dosed women with beta-carotene that had the 2 forms of the isomers, and we looked at appearance in milk. And what we found is that even the beta-carotene trans was better absorbed than the beta-carotene 9-cis, but tissue uptake was about the same. So that was a fun experiment. And that was done with Jan Chin, who was in our lab at that time, but now works in the NEL. And she was really helpful in getting this work done. All right, so that's some work I had been doing with beta-carotene. And then something happened. And there were 2 publications on supplementation with whopping doses of beta-carotene. And they're the alpha-tocopherol beta-carotene trial and the beta-carotene retinol efficient trial. And the reason those studies were designed was because at that point in time, it was realized that beta-carotene has, or vitamin A has effects on cell differentiation, and epidemiology related it to cancer. Those people that are eating a lot of beta-carotene in their food and had a lot of it in their blood had less risk of cancer. So the idea was, in these studies, if we dose people who are at increased risk of cancer, let's see what happens as far as decreasing the risk of getting cancer. So they did that, and these doses were beyond what you get in diet, and the subjects that they chose were at high risk for getting cancer. So they were heavy smokers, asbestos workers, and they did this trial, and guess what happened? They got more cancer. So that was bad. That was really bad. And so that kind of was a game changer for me as far as studying beta-carotene. And it was a game changer for Xiangdong Wang as well, because this really turned his work into a different direction by studying how that happened. So when it's his turn to get up here and talk about his 25 years here, he can tell you how his story ends with this work. All right, so I'm not studying beta-carotene anymore. I'm going to go to lutein. And not all carotenoids are alike, okay? So beta-carotene, provitamin A carotenoid, pretty lipophilic, lutein, no vitamin A activity, and it goes to tissues, different tissue distribution than you find in beta-carotene, which is interesting, and hopefully you'll understand why that's interesting. So now I'm studying lutein. And why, so the first study I did with this is another MRU study where we looked at dietary modification of macular pigment density. Macular pigment density, what is that? Well, what it is this is your macula is the center part of your retina, all right? So I'm looking at you right now through my macula. So because it's the center part of your macula, it's the most vulnerable part of your retina. So with aging, as we're all doing right now, and decades of light exposure and that direct hit of light on your retina, on your macula, it's the part of your retina that's most vulnerable to damage as you age from all that light exposure. Good news is, here's the light coming in. See that yellow pigment there? That yellow pigment is lutein and its isomer zeaxanthin. And they get concentrated in the macula to about 500 times more concentrated than any place else in the body. So that lends some biological plausibility for it being important. And if you look at it, it's right there, right in front of those photoreceptors. So as the light comes in, it grabs that light so it can't go on to damage the photoreceptors that are underlying the macular pigment. And that's a good thing. And there's a lot of different kinds of evidence to support that you really want more macular pigment because it's an antioxidant, it's a blue light filter. People with high levels tend to be at decreased risk of age-related macular degeneration, which is one of the major causes of visual impairment among the elderly. So we really want high levels of this. So we looked at what happens if you have a simple dietary modification to increasing that. Can you increase it? And that's what we did. We fed people, and some of them are in this room, we fed them 2 ounces of cooked spinach for, how long, 12 weeks? So that's this much spinach. It's like you buy a 10-ounce package of frozen block of spinach. That'll last the week. That's 2 ounces about. And what we found was that with that modest dietary modification, we could increase macular pigment levels, in a couple months. And so this was the first report of how a dietary modification could increase macular pigment levels. What we also found is similar to what we found with beta-carotene a few slides back. There were some people that didn't respond as well. So we found that too, that some people did okay as far as a serum response to the dietary intervention, but they had no changes in their macular pigment. And there was one person that really didn't do anything. So people are different. Not everyone handles a carotenoid the same in terms of absorption, transport, and uptake into the retina. So that became interesting to me, is why are people different? And so one place that we decided to look is like what's going on as far as the rest of the body, what's going on below those eyeballs. And this is work that I did with, along with Kyung Jin and Xiangdong Wang, with Carmen Castaneda, who was at that time in the NEPS lab. So now I'm working with another group where we did some tissue biopsies. We did adipose tissue biopsies to look at carotenoid concentrations there and how they relate to macular pigment. And the reason for wanting to look at adipose tissues, that's where most of your carotenoids are going to be. They're lipid soluble. It's going to be the major storage site of these pigments. And does that somehow compromise what's going to get to the tissue of interest, the retina? And so that's work that I did with a schoolmate of mine in Wisconsin, Julie Mayers, where we actually looked at that. In the sub-study of the Women's Health Initiative, we looked at what were the predictors of macular pigment in these women. And there were 4 things that popped out that were independently related to macular pigment. And they were dietary lutein, which makes sense. If you don't eat it, it's not going to get to your eye. You have to eat it. It was diabetes. So women with diabetes had less macular pigment. And that probably is related to an oxidative factor. Oxidative stress will decrease your status of lutein. But what we also found was that BMI was an independent predictor of macular pigment. So more body mass fat, less macular pigment. But it wasn't only the amount of fat, it was where it was. So it was more that was around the abdomen, less was in the eye. So the distribution of body fat made a difference as far as how much macular pigment these women had. So that was interesting because of a study that we did that actually looked at the distribution of carotenoids in different body fat sites. So we looked at the abdomen, we looked at the buttocks, and we looked at the thigh. And what we found was that in the abdomen, for both lutein and its isomer zeaxanthin, and usually when you hear a talk about lutein, you hear these 2 carotenoids together, because not only are they isomers, but they're commonly found in the same foods. So we looked at those, and they're both macular pigments. So we looked at that, and you found that about twice as much lutein was found in the abdominal fat than the other fat sites. So that was really interesting when we think back to the study where, more abdominal fat, less in the macula, thinking that perhaps if you accumulate it here, it makes it less available to the eye. So I was thinking about that as well and trying to piece it all together. And so I did in a Nutrition Reviews article that looked at all these connections among lutein status, obesity, and of interest because of lutein was age-related macular degeneration. And there's a lot of different reasons to believe that obesity, macular pigment are all related to risk of AMD, and so I go through that in this article, and I won't go through it today because we don't have that kind of time, and I have many more things to say, but if you're interested, there's an article in Nutrition Reviews that talk about these connections. All right, so that is macular pigment, and we're really interested in how are we going to get more macular pigment, because there's a lot of compelling data to believe that more is better. And up until this point, people were thinking about fruits and vegetables, carotenoids, fruits and vegetables, but no one really considered eggs. And so this is a study I did with Helen Rasmussen up in the MRU, the research dietitian there, where we actually looked at eggs as a source of lutein. So now think about an egg. Think about an egg yolk. It's yellow to orange, right? And why do you think it is? There's only one answer you should give me, and it should be lutein. Egg yolks are that color because of lutein. So the idea was, eggs, are they going to be a good vehicle for lutein? And the idea is probably yes, because they're not like spinach, they're not like kale, where you got your cell plant wall and you have to juice it, you have to cook it to break that wall, to allow those carotenoids to be absorbed. Well, lutein's just swimming around in the egg yolk, in those phospholipids. The assumption is that that's going to be a really good vehicle for lutein absorption. So we did that. And so Helen helped us design a good meal for these subjects to eat, where we balanced the protein, the fat, and all the macronutrients, and looked at the absorption of lutein in egg and compared it to spinach, which is where most people are getting their lutein, and supplements. And what we found was eggs were the big winner. Eggs, the bioavailability of eggs was about 3 times more than spinach, than lutein contained in a supplement, whether it be in the free form or in the ester form. These were all about the same. So that was really great. So now we have a food vehicle that wasn't really considered for lutein, that wasn't being considered before. And at the Aging Center, this was good news, because when you think of eggs, they're really a pretty good vehicle for nutrients. I mean, they got the perfect protein, they're digestible, they're pretty cheap, and so now we can think of them in terms of lutein, as being a source of lutein. So that's lutein, and so during this time as well, I was getting involved with other labs, and I had the good fortune to work with the cardiovascular nutrition lab with Alice, and because of our carotenoids and the interest in antioxidants and all that, we did some work looking at soy and proteins and isoflavones, and found that they didn't have much effect on antioxidant function. I also did some work with Joel Mason's group and Jimmy and Choi, where we looked at non-smokers and smokers and looked at carotenoid, retinoid, and tocopherol status. And what we found, to no surprise, is that smokers, they have less carotenoids in their serum that wasn't accounted for by diet, which makes sense because smoking is a pretty oxidatively stressful situation, and carotenoids don't like that. So that was work I did with them, and that was good for me. So everything I've been talking up to this point were carotenoids, its status, antioxidant function, bioavailability. And at this point in time, things got a little bit different for me, where I started looking at cognition. And this work was done with the neuroscience and aging lab, and at that time, Aaron Trone was here and helped me with this manuscript. And what we did was we looked at a double-blinded placebo-controlled trial involving lutein and DHA. And the reason for this study design was because the original primary outcome of interest was visual function. So remember, I was talking about macular pigment, I was talking AMD, where we were really interested in looking at these bioactives and the effect on visual function. But because we were looking at DHA, we wanted to look at cognition as well, because there was a lot of evidence to support DHA being important for cognition. So we had these 4 groups of older women who were recruited for their visual function. They weren't recruited for cognition. That was a secondary outcome. And the study involved placebo, so there's a placebo for both lutein and DHA. There was a lutein alone at a dietary level. It's 12 milligrams a day, which you can easily get in diet. Remember that 2 ounces of spinach? That's about that same dose. There was 800 milligrams of DHA, and there's a combination. So remember, it's a double-blinded. And what we did is when we looked at the data and unblinded ourselves and looked at cognition as measured by executive function, we found that all the interventions did better. So women improved from baseline, whether they got the lutein alone, the DHA alone, or the combination. And that was a surprise, because there was nothing before that ever hinted that lutein was going to be important for cognition. So I began to start thinking about this, because we found it in other measures where the combination of lutein and DHA did better. And we found it in another measure as well. So now I'm thinking about, why is that? I was thinking about what can I do to see if there's something going on with lutein and cognition. But while I'm doing that and designing those studies and figuring that out, I got involved with the nutrition epidemiology lab. And in this study, we looked at dietary patterns and relationships with carotenoids and tocopherols and concentrations. And this was with Katie Tucker, who was here at that time. And what we found, to no surprise, that a dietary pattern that included snack foods, fast foods, pop, and meat were low in carotenoids. So when you think about where you get your carotenoids, that's not going to be a surprise. And then I had some work with Jeff Blumberg, whose lab I'm a part of right now, where we looked at plasma carotenoids and found that the xanthophylls, which are the more they have OH groups, as opposed to the carotenes, which are more lipid soluble, that they were related to markers of oxidative stress, where the xanthophylls were and the carotenes weren't. So again, not all carotenoids are alike. And I also worked with the Vision Lab with Fu Sheng, where we looked at another population. The work with Katie Tucker was in African Americans, and with Jeff it was cancer patients, and now we're looking at a Chinese population. And we're looking at relationships with carotenoids and age-related macular degeneration, and we find the same thing. We found that zeaxanthin, the isomer of lutein, was related to a decreased risk of AMD in Chinese. And that was the other thing we found in this study is the Chinese had a much higher serum levels of carotenoids than we find in the US, which isn't too much a surprise when you look at what they're eating. And then I did more work with the Vision Lab, as well as Guang Tang, who is retired now, but still, very much a part of the work that I had done, and Alan Taylor, where we did some cell culture work, and we looked at some mechanistic ways that lutein may be working to protect vision. We found that it is involved in modulating the immune response. So that was in cell culture. So at this point, students, you should be getting an appreciation of the diverse amount of tools we have in this building. So I talked about epidemiology, I talked about clinical studies, I talked about some observational studies, and about cell culture. So under this roof, we're able to do a lot of different kinds of experiments to answer these questions. So I've been really happy to be a part of that diverse, those diverse study designs. But we're doing a lot of good work, and a lot of that gets out there. But the next thing I want to talk about is how we translate the science so people can use it. So this is work that I did with Helen Rasmussen, and this was at a FENCI study where we had a booth where we were really talking about the importance of lutein and AMD. And so what we did, Helen and I, we wrote an article that really looked at the nutrients that are important for aging. And how do we How do we give people an understanding, an easy understanding of what nutrients, what foods are important for visual health? And this is what we came up with. So it pretty easily, quickly communicates vision, but also the nutrients that we believe are important. The nutrients we picked are those that had been shown in a double-blinded, placebo-controlled trial, the age-related eye disease study, where supplementation with antioxidants actually decreased progression of AMD. So we got those nutrients in our chart, as well as foods from which they come. So we really wanted to communicate to people in an easy manner how to really, make their diet help out their eyes. So that was fun. Okay, so now we're getting back to cognition. Remember I talked about it earlier, and I'm thinking about what kinds of studies to do to really see if lutein's making a difference. And I had the good fortune to be able to work with Rohini Viswanathan on some of these studies. And so one of the first that we got published was looking at macular pigment and its relationship to brain levels of lutein and zeaxanthin. And what we found was they're actually correlated. So more macular pigment, more lutein and zeaxanthin was related to more in the brain. And this work was done in primates, so we used monkeys. And then we confirmed it in humans, where we had matched retina with brain tissue. And look at that. That's like beautiful. It's beautiful. You don't see that a lot in human old people. These are old people. And when you think about it, though, it's really not too much of a surprise, because when you think about getting lutein from the food, getting it absorbed, having it travel through, and going past the blood-brain barrier, if it's going past there to get to the retina, it's going to go past there to get to the brain, right? So that's not too much a surprise, and that's terrific, because now we have a biomarker of brain levels of lutein. And macular pigment levels are pretty easily done. And if you come up and ask me, I can do yours in 5 minutes. We go to the 11th floor. It's a simple test. And if I do that, I'll get an idea of how much you have in your brain, which you may or may not want to know. But anyway, why do you want to know? Well, this is why. And Tammy will tell you why. Tammy Scott and Rohini and I have this publication where what we found was we had access to brain tissue for which there were pre-mortem measures of cognition. So that was really amazing. And so we had those measures, and what we did, we also had serum. So remember I told you that lutein and zeaxanthin get preferentially taken up into the macula, right? They're the only carotenoids that go there. So there's preferential absorption. The same thing's happening in the brain. So these are decedents for which we had serum and we had brain. And if you measure carotenoids in serum, that's going to be a pretty good biomarker of what they're eating. And so what these people are eating, they're eating a lot of beta-carotene, significantly more than any other carotenoid, right? But when you looked at matched brain, lutein was the one that was the major carotenoid. So just like in the macula, the brain preferentially takes up lutein. Is that important? Well, we think it is, because when we looked at measures of dementia, this is a dementia scale, for which one is cognitively intact. Here's some mild memory loss and then some mild cognitive impairment. The scale actually goes to 7, but we didn't want to look at 4 through 7 because that's dementia. And usually once people get into stages of dementia, nutrition doesn't have as much of an impact. So we were really interested in the target population where nutrition could be important. And when we did that, look at that, lutein levels in those that were normally attacked, much, much higher, significantly higher than those with mild cognitive impairment or memory loss. We didn't see it for zeaxanthin, we didn't see it for beta-carotene, we didn't see it for alpha-tocopherol. And when you think about alpha-tocopherol, what do you think about? Antioxidant, right? So here you have alpha-tocopherol with levels like 1,000 times more in the brain, but not related to cognition. So the idea here is what carotenoid is this? Lutein. If lutein is involved with cognition, it may not be as an antioxidant, maybe something else. So that's lutein. We did this for other measures of cognition, and the same thing. Lutein seemed to be the one that was doing something. And it wasn't so much how much was there, you know, compared to the other carotenoids, because cryptoxanthin was there in levels just as about the same. We didn't see the same thing. So it seemed to be unique to lutein. So, macular pigment, so we found that in brain tissue, and I told you macular pigment's a biomarker of what's in the brain. And so in this study that I also did with Tammy and Rohini, we found that in a cross-sectional study, people with higher macular pigment had better cognition. So we know why now, right? Because macular pigment was a biomarker of what's in the brain. So in this study, the presumption is these people had more lutein in their brain, which was related to cognitive function. And so we have this work with macular pigment and relationships to cognition. There have since been two other labs that have found the same thing. So we got some consistency there on that. Oh, and this is just to prove that we found relationships between macular pigment and a variety of measures of cognitive function. Okay, so that's what I've done up to then, and so now we're in 2014. What am I doing now? So we're trying to figure out what lutein's doing. So here's Emily Mann, who's doing that. So hopefully we'll be hearing about that. And what she's doing, you know, I told you that there's a lot of lutein in the brain. It gets preferentially taken up. And that's one thing, but we want to know where is it going. We want to know what is it doing. And so we're looking at where it's going on a subcellular level. So Emily is fractionating out brain tissue in rhesus monkeys and looking at, you know, what membranes it's going to. And then she's relating that to some measures of cell viability. So this is her dissertation project. And she's got her own cluster going on because she's working with the antioxidant lab, which she's a member of. She's working with the neuroscience lab. She's working with the cardiovascular lab, doing a lot of these measures to look at relationships with the amount of lutein that she's finding in these subcellular membranes. I'm also doing work now with Nirupa and Jakob, where we're looking at nutrient biomarkers and their patterns and relationships to cognitive function in healthy older adults, because it's not just about lutein. There are other nutrients that we think are important for cognition, and we're looking at, you know, combinations that really prove to be of most benefit. And that's a lot, NEL is helping us with those measures. So we're looking at B vitamins, we're looking at fatty acids, we're looking at choline, we're looking at vitamin D, we're looking at a lot of different things. And then with Oliver, as well as Rohini and Tammy and Emily and other people, we have a study that we completed this summer that involved an avocado intervention. So avocados, they actually are a nice vehicle of lutein. There's not a lot there, but because of that vehicle, that fat that's there, it really gets well absorbed, as you see in this slide. So this was an intervention that Helen helped us design, because she designed a lot of the foods that these people eat, because they had to eat an avocado a day for 6 months, or else they got a potato or chickpeas. And look at that. They had significant increases in macular pigment. We found the increases in serum as well, and that was terrific. And in fact, when we looked at the changes in macular pigment, we found that there were measures of cognition that were improved as well, and this was in healthy older adults. So this is work that we'll be presenting at EB in the spring. So that's what I'm doing now. So, and then we just submitted something with Paul Jakes. It was a meta-analysis where we looked at dietary cholesterol and cardiovascular disease. Had nothing to do with carotenoids, but it really was good for me to go through this exercise of what's involved in a meta-analysis, so I really appreciated this work. So, what did I tell you? Well, I got here in 1988, and at that point in time, when you look at Medline and you punch in the keyword for carotenoids, beta-carotene, lutein, these were the publications that were going on. And today, a lot more has been going on over those past 27 years, such that you have 10 times the amount of beta-carotene publications that happen and about the same 10 times the amount of lutein. So a lot has been going on. A lot of that work has been great, and in part because of some of you that are sitting in the audience. But I recognize some of the scientists, some of my coworkers that had been involved in these studies, but also a really important part to all of this were those people involved in the MRU. So we had our recruiting, our nurses, our dietary, because, you know, I'm working with lutein, and when you work with lutein in the eye and cognition, you can't work with a rodent, you can't work with a guinea pig, because of how they handle carotenoids. It's very different than a primate. So I have to use the human as my guinea pig, and I only can do that with the help of all the staff that we have in our MRU and our staff assistants who have really, really helped. So they really need to be thanked. So that's the end of my story. So I'd be happy.
So, well, I still can remember my question. If you look at the brain of, like, say, Alzheimer's patient or something versus a non-Alzheimer's patient at the same age, do you see any difference in any kind of nutrient concentrations, protein or any other?
We actually did do that. And I think the problem we had is the composition is so different of an Alzheimer's brain that we had trouble with what the denominator was. And in fact, in our Georgia centenarian study, we had, you know, normally intact and mild cognitive impairment, and made it Alzheimer's. It kind of fell in the middle. So we had a hard time understanding that. The interesting thing about those Alzheimer's brains, though, is that the vitamin A content was really high, and we found that it was related, you know, to the provitamin A carotenoids, and so there was a story there that we really haven't teased out. But again, you know, we were really more interested in looking at those for which, you know, nutrition would make a difference, and the Alzheimer brain was just really difficult for us to figure out what those data meant because perhaps of the composition of that brain.
Very nice topics. So if you look at the structure of all the carotenoids, particularly xanthophyll, that include lutein, beta-carotene, and zeaxanthin, the chemical structure are very close.
Right.
Very similar. So now, you know, you show clear that lutein is unique. Is there any idea about potential mechanism? You know, how this lutein is so unique in terms of compared with the other?
Well, we don't know, and I think that's some of the work that Emily will be doing that may help us understand what is it doing that the other carotenoids. Well, one thing about lutein, when we're looking at those subcellular membranes, we'll look at the whole tissue, and you'll find carotenoids in the whole brain tissue. But when you look at the membrane, it's really just lutein. Sometimes you see zeaxanthin, but you just see lutein there. So that makes it unique. And there's a lot of reason to believe, you know, being embedded in that membrane is going to have functional consequences. So maybe that's it.
Very nice talk. Do you have any reason to believe that there's a visual component in these cognitive function correlations or associations that you've seen? In other words, is it truly all cognitive, or is there, because much of the cognitive assessments are visual in nature, do you think there's a component to
Right.
do you suspect any role in those?
Well, for one, it's really hard to tease out vision from cognition, and that's a real hard thing to do. And yeah, sure.
Some of the tests weren't visual tests, so.
Yeah, I know some of them
Right,
were.
so they were
Yeah.
still related to cognitive function, even if they were not visual tests. So although there may be some overlap there, it doesn't seem to be all the only thing that's going on.
Yeah, I didn't think it was the only thing, but okay. So
Yeah.
very visual.
Yeah, it's really neat though, because vision, you know, the eyeballs are just a part of the brain, and it's all, you know, you see a lot of the same risk factors for AMD as you see for cognitive function. Joel?
So with beta-carotene, we, at least in regards to beta-carotene in the cancer store, we kind of learned that more isn't necessarily better. Might the same be true for lutein and cognitive health? I mean, is there a limit beyond which we should not be pushing lutein levels in the macula or in the brain?
I always like to say work with a dietary level, which, you know, you can easily get 12 milligrams of lutein from the diet. And the 10 milligrams is what was used in the AREDS-2 trial, which found to be beneficial for decreased progression to AMD. But with that said, there are populations in the world where they eat 40 milligrams of lutein a day from their diet without any problem. I don't think you really need that much because I always worry about competition among the carotenoids. Lutein has its function and cryptoxanthin may be doing something, and they compete. They compete for transport, absorption, tissue uptake. So I don't think, and I think Xiong has looked at lutein and toxicity.
The reason because the beta-carotene trial really is a very bad representation of the carotenoid. But remember after the interaction between the bismol and the beta-carotene concentration, in the other population, really not much evidence to show it harmful. So I think they, in terms of lutein and bismol, there is no data.
Helen?
Is there any data about the, you were talking about the Asian, the macular, increase in the Asian population. Is there any sort of experiment being done on the people that are in the highly polluted areas with the antioxidants?
Oh, not that I know of, but that's a really good question because that's oxidatively stressful.
Would that be maybe why they have a higher level of macular? Or,
They had high levels of serum carotenoids, and so we found that zeaxanthin was related to decreased risk. AMD in China only recently has been recognized as, they've only recently been looking at that, and it's been recently diagnosed, and it's becoming more and more of a problem. And China actually has a recommended dietary intake for lutein. So.
I have a question.
Okay.
So you talked about lutein and zeaxanthin in the retina, but you didn't talk about mesozeaxanthin. Do you think it's an independent player?
I think mesozeaxanthin, there's a lot of discussion right now. So I told you there were two carotenoids in the macula, lutein and zeaxanthin. Well, actually there are three, and one is the mesozeaxanthin. And right now, the discussion in the industry is which is more important. Is lutein more important? Is zeaxanthin more important? Is mesozeaxanthin more important? And depending on who's selling the product, they'll give you a different answer. So what NEI found is that 10 milligrams lutein, 2 milligrams of zeaxanthin was beneficial for those with low intake. No mesozeaxanthin in that product. You don't find mesozeaxanthin in food. You find it in salmon skin. And you'd have to eat 200 pounds of salmon skin to get the amount that was beneficial in the NEI's trial. So it's not a dietary component. But some people are thinking it's important because you find it concentrated in the macula, so macula wants it. But I'm saying it gets it there because you convert lutein to mesozeaxanthin, and we showed those studies in our monkey trials that the source of mesozeaxanthin in the macula comes from lutein and not from diet. So you might not want to supplement with it because it's not something normally found in your diet. And when I talk about competition among these carotenoids, if you're going to supplement with something that your body's not used to seeing, such as mesozeaxanthin, is it going to compromise what lutein's doing, what zeaxanthin's doing, particularly for a young child, because you got neural development, you got visual development in that first year of life. So if you're putting something in that brain and eyeball that's not used to being there, could that have impact on that development?
Any other questions?
So the lutein convert to mesozeaxanthin, it's by the enzyme or it's
We don't know. Paul Bernstein's looking at that, and you think it might be involved with that binding protein, but he's looking at how that's done. But it's very specific to the very center of the macula. You don't see it a step this way, you don't see it a step this way. So you don't find it in the brain. We haven't found it in the brain, so I wouldn't supplement with it.
Thank you very much.
Thank you.